Let's get started. So I know that we talked about the photosynthesis, but let's kind of track back a little bit into I'll continue on with the photosynthesis. Let's take a look at, guys, the cellular respiration. Okay. So let's talk about respiration first, and then move down to the photosynthesis again. Okay. So in regards to respiration, cellular respiration. So the purpose is to produce or convert glucose, or it doesn't have to be glucose. I mean, but for the AP, I mean, they've kind of restricted to glucose, or you can say like any monosaccharide or any sugar, okay, and convert glucose or utilize glucose to produce ATP. Okay, so that's basically one of the things that you have to know, guys, by the way. Okay, glucose to ATP. So we talked about what ATP is. Okay, adenosine triphosphate. So I'm not going to repeat that. Okay. So let's kind of discuss again. Okay, why they need to produce ATP, right? So, okay, what's the purpose of having ATP? You know, it's one of the stuff that you can ask yourself. You know, so when you're taking the like easy bio, that whatever you guys did. Okay. So that was like, oh yeah, ATP, they used to call this like the currency of energy, you know, for the cells, and that was it, you know. And then you have the ATP for the source of energy, blah, blah, blah. So, but I think we talked about, you know, more specifically how ATP can provide, you know, the energy for the entire process, right? But, so how do they do it? So one of the stuff kind of tracking back over what we discussed as well. For example, A turns into B, for example. This is an end-organic process overall. So just to kind of let you know, right? So this process is the inorganic process, right? So inorganic process, as you know, is what we consider as like, you know, delta G is equal to positive value, right? So if delta G is positive, if change in free energy is positive, that's what we call the inorganic reaction, right? Which means like this is in chemistry perspective as well, guys. It's a non-spontaneous reaction. It doesn't happen on its own, right? So because of the fact that it doesn't happen on its own, what happens, guys? Yeah, it will never occur on its own. So which means what? They can push the reaction with the assistance of ATP. So meaning, guys, so what they're gonna do is what, guys? Okay, so basically, you know, they're gonna break not really break, but they're gonna use the ATP, right? So now A plus ATP, okay. So ATP will be utilized, okay, with A, and then turned into what, guys? A now will be phosphorylated, okay, and then you have an ADP. So this process, again, you gotta understand, guys, ATP, all the stuff we talked about is what, guys? Why ATP is being utilized as a currency of energy is because ATP is what? I mean, they have a relatively unstable, like, you know, phosphate group of them, right? So which means if you look at it, so phosphate, 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 right? So these are triphosphate, right? So these are all negatively charged, or even like, or they have a highly electronegative, okay, oxygens on them. So one thing to know, guys, what you should know is what, if they have a negative charges all lined up like this, they tend to what? They tend to repel each other. Okay, they tend to push away each other, right? So because they do that, what happens, guys? Yeah, because of that, the bond is what? Relatively unstable. So instability means what? Instability means it has a high potential energy, okay? So now if this breaks off, okay, this is a third phosphate, breaks off, what happens, guys? You only have two phosphates on them, and one phosphate broke off, which means what? This is more stable. Okay, so because of that, guys, means ATP breaking into ADP and phosphate, okay, that gives them what? More stability. So, you know, so this entire process is what, guys? Now exothermic. So this is like where delta G is what? Is more negative. It gives off heat. Okay. And keep in mind, guys, when one of the stuff that we also talked about as well, when something gets phosphorylated, like this, right? When something gets phosphorylated, which means, so basically what happens, guys? Yeah, the phosphate group was here, but now they put it on this guy. There's an enzyme. You have to know this guy. There's an enzyme that does that, which is what enzyme called kinase. There's an enzyme called kinase. It's a kinase, right? So what happens, guys? They take the phosphate group out and put it into what, guys? One of the substrate, right? So what happens, guys? Yeah, there's an enzyme that does it. You have to know that, guys. At least the term kinase here, and this becomes what? So when a substance like this gets phosphorylated, what happens, guys? They become more unstable. They become relatively high free energy. Okay, and then utilizing the energy of this. Okay, so now A with the phosphorylase, what? Yeah, they're relatively unstable. What happens, guys? They easily can turn into B because they have a greater unstable free energy in what? Phosphate comes out. So they make it into like a step here, okay? So because of this, because they use ATP for this process, what happens, guys? Yeah, it becomes relatively what, guys? The process can occur with the assistance of what? Kinase and ATP. So for your cellular activities, right? You know, like, okay, so ATP, you understand where this will be utilized, okay? So so high level is what? To utilize, something like ATP for what? To run a reaction that's what? Endergonic reaction. It doesn't happen on its own, so you need to have a system for producing something else. So that's the reason why they have to produce ATP, okay? So you know. So the question for today becomes, well, how do they produce ATP? Okay, how can they convert the glucose to ATP? So the question is, guys, so every cell that you know, right? Everything that you know, which is prokaryotic cells, eukaryotic cells, they all have to convert this to this. Okay. ATP. So we have to discuss, guys, how they do this. And this is something we already discussed. Okay, this is like nothing new for you guys, right? I think I did. Guys, I mean, keep in mind, guys, like, get your COVID shots, okay? So, dude, I don't care, like, and not that I don't care, but I don't work for the state or the city. I mean, dude, like, it's free, okay? You don't need an insurance for this, and you guys are probably having insurance on your own or something. I don't know. Get your COVID shot, guys, if you haven't. I think 12 and up. I tell you guys, I was telling some of the kids here. I have a coworker, like, I tell you, man, like, I was like shocked when I heard this, right? I have a coworker who's like around my age, like, 21, I'm just kidding, like, early 30s, 30s, okay? I tell you, man, like, he got his lung out. His lung is gone, bye-bye. They took his lung out because he wasn't responding to the treatment at all, okay? And then they put an artificial lung, but he's still not awake. He's still, like, not awake. He has a kid, he has a wife. I mean, dude, like, you can't, I don't know, you can't let your life go like that. I tell you, but I don't know if it was vaccinated or not, guys, but that's not a way to go, guys, by the way. So it's too young. You guys are like, what, like 17 or 16, something like that, I don't know. So you guys have at least like 70 years or more to go. So don't end your life early, guys. Get your COVID shots, okay? And don't take your chances, okay? All right, so and some people truly don't believe. I don't know, like, dude, like, even my brother, like, he's like, oh, dude, like, oh my god, like. All right, so let's take a look at it, guys. So one of the homework question had this, okay? So I didn't really check if we did it or not, guys. I'll check when we go over, guys, by the way. So how glucose, okay, comes into a cell, okay? So how can glucose come into a cell? Glucose is what? So when you draw the molecule of glucose, okay, so glucose has a lot of these hydroxyl groups on them, OH groups, right? They got a lot of these guys on them. Think about it, right? So these hydroxyl group can what? That's what makes them very what? Hydrophilic. Okay, they're very hydrophilic. They're polar molecules, right? Just right now. So, I mean, it's not a large molecule. Okay, water, like, this is not a large molecule, but the question becomes, right? Can they come into a cell by diffusion? That's a question. That's a question you can ask yourself, right? Can they just come in? Like, is that something they can do? Is something you have to ask yourself. So, what is the first thing you have to ask yourself, guys? Can they come in, okay, through the cell? So what's the first thing you have to ask yourself, guys? So dude, like, okay, first thing is it's a polar molecule, okay? So we understand, guys, okay? So the cell membrane, as we know, is what? They have a hydrophobic layer zone, right? Like, you guys probably know that, right? So, which means, what can just come into a cell by diffusion process? What? Very small, tiny hydrophobic molecules such as, like what? And like N2, like O2, CO2. They're very tiny molecules like N2, O2, and H2, CO2. They can come through diffusion or steroid molecule, right? But definitely not glucose. Glucose will not be able to come in by diffusion. That's one of the stuff that we talked about. Okay, so keep in mind, guys. Listen very carefully, guys. Water H2O. So this is, so I had an issue with H2O with one of the teachers, by the way, okay? So water, well, this water can come in by diffusion, okay? Diffusion, but at a very slow rate. Because why? Water is very polar, okay? So, dude, I tell you, man, like, if this is like a SAT2 class back in time, right? So SAT2, the exam back in time, they think water can come in by diffusion, okay? Which means like, yeah, they pass the membrane, like, yeah, that's how they come in. AP, absolutely not true, okay? You can't say that anymore, guys. Because the water is so slow coming in, because this is very polar and there's a zone of hydrophobic, they have to come through, this is something I mentioned, guys, okay? They have to come through a channel, okay? Protein channel called what? Aquaporin. You have to know this, okay, so you know, for the EP. Aquaporin. So aquaporin is something that's around the cell, okay, for every cell. It's like, dude, there are a lot of these guys, aquaporins, okay, the cell membrane. So because the water cannot come in or go out at ease, okay, what happens, guys? There's a channel, protein channel, the transmembrane integral protein that what? That lets the water to come in, okay, by facilitated diffusion, right? That's what aquaporin is. So if you truly think, guys, how does water come into the cell? Is what, guys? Yeah, basically what, guys? Facilitated diffusion, okay? They cannot just diffuse into a cell or just go out, okay? They can't do it because what? I mean, they can, but at a very slow rate. At a very slow rate, okay? So going back to glucose, what happens, guys? Yeah, they can't come in, okay? They have to come through a little channel, okay? They have to come through a channel, right? So, you know, again, when there's a lot of glucose available, then what happens, guys? Yeah, like, they kind of knock on there's insulin, there's a whole different chapter on that, guys. It's make the cells too aware there's glucose around, and what? Open the channel to let the glucose in. Okay, so once the glucose comes in, the question becomes like, what are we going to do with this? That's basically what this chapter is, and how they convert this into ATP. So how do they convert this guy? Not really like turning into ATP, but convert this guy into ATP. That's what the question, entire chapter will become, okay, for today. And you know, the photosynthesis also, guys. Okay. So one of the things that we talked about, or I mentioned, guys, again, is where is dude? Like you hear this, like there's the energy here, right? So where's the energy in, guys? What are you doing? Where's the energy in? Yeah. Yeah, like dude, like it's what? The electrons in the carbon. Okay, the electron in the carbon. That's where the energy is at. So your teacher, I mean, so do you have to really know this, guys? I don't really. So AP, like the questions, they never really ask you directly. Okay, so yeah, guys, tell me where is the actual energy in? I mean, they don't really, you know, talk about that too much, guys. By the way, your teacher might say there's the bond between carbons. You know, they might say that, just so you know. Do I agree? I don't know. So I get it, but okay. So, but yeah, it's what the carbons have electrons in them. Okay, so yeah, the electrons might be, you know, that's bonded to the other stuff. Understandable, so, you know, but again, it's electrons in the carbon. That's what truly gives them the energy for the whole what process to turn it into ATP. So what's the first process you have to know? Okay, so what's the first process, right? Is one of the stuff you have to know about the glucose entering the cell. The first process is called glycolysis. So glycolysis is the first step, okay, for all. Like, you know, for every, it's like, dude, like this glycolysis process, just so you know. So we haven't talked about evolution. So I don't know, yeah, a lot of Jericho folks here. So the first thing when you guys go to school, you guys will talk about evolution. So I don't know, maybe, I don't know, the last class that we'll have in like a couple weeks, I might want to, I don't know, not do cell cycle, maybe cover evolution for you guys. I mean, so maybe, you know, but evolution perspective, okay? Glycolysis, so this is how they say it, right? Glycolysis is a very conserved process in an evolution perspective. Again, what the hell does it mean is what? It's a very conserved process. Means, so, you know, in bio perspective, it speculates what, guys? Okay, so we started from a very small, you know, prokaryotic cell, okay, into what? I mean, dude, like, you know, I'm not very, again, religious, by the way, But now what we got human, like dude, like we got mammals, we got planes and everything, right? So they say, okay, so from a very tiny, like first living organism, whatever that was, okay, and now it all evolved to what, guys? All the crazy stuff that we have, like millions of different species that we have. So, but when they say it's a very conserved process, means what, guys? Every one of these guys can perform glycolysis. You know what I'm saying? So, which means that every cell that's out there, they can do this. At least glycolysis, okay? Because what? That's the very fundamental process to produce ATP, okay, from, you know, simple glucose molecule, okay? So what the hell is glycolysis, okay? So what do they do it? So I tell you guys, okay? So AP perspective, okay? Glycolysis is not something that you will have to memorize at all for AP. Like, okay, how much do I have to know about glycolysis of AP? Not a lot, okay? You just have to know what the hell happens, okay? You just, oh yeah, like what goes in, what happens, and what comes out. That's all you have to know, guys, for AP. So they don't go too crazy on this. I mean, you know, they talk about this a little bit of evolution, just like what I mentioned, guys, a very conserved process, blah, blah, blah. So let's take a look. What happens in the first process of glycolysis? So glucose went in. Keep in mind, guys, this happens in cytoplasm, right? So where this happens, guys? Cytoplasm. What's cytoplasm, guys, again? Cytoplasm, what, guys? So if it's eukaryotic cell, okay, the space between the nuclear membrane to what? The cell membrane. So every, you know, location or region between the nuclear membrane to the cell membrane is what we call cytoplasm. Okay, this is where glycolysis occurs. So if you look at this, let's see what happens. Okay, so glycolysis is a 10-step process. Okay, there are 10 steps to this. 10 steps. 10 steps. Okay, which requires like 10 enzymes. Okay. Do you have to know all the steps, guys? You don't have to know them. For the AP, you don't have to know it. Okay, so for students who goes to some of the crazy school, I don't know, like some weird school, they probably make you aware of some stuff a little bit more than what you need for the AP. Okay, I haven't seen them. Okay, but if you guys go to college, okay, then you have to know the steps. Okay, just so you know, like for example, if you guys go to college and take biochem class, that's a 300-level class, by the way. So I'm sure, or you know, they will probably make you memorize some of the steps here in the glycolysis process. So what happens to glycolysis? Okay, glucose goes in. Okay, so as you can see from the name, glycolysis, what happens, guys? Glucose splits. Okay, glucose split up. The glucose go in. What happens, guys? So I mean, this, even what I'm mentioning right now, guys, might not be something your teacher will mention. Okay, but maybe like you'll get a handout, maybe, you know. Glycolysis overall have two phases, like, you know, two phases of the glycolysis. You know, just so you know. So there's energy investment phase. Okay, the energy investment. Okay, and there's energy payoff. So investment and payoff. Okay, so which means, okay, so you want to make some money. So dude, I don't know how you guys are open to the, like, you know, guys or whatever, to the like investing. Yeah, some people like put in like, oh yeah, I bought like a, you know, 100 Dogecoin like, you know, 5 years ago. Now it's like, oh my God, you know. So that's pretty much where this goes, right? So if you don't like, okay, so if you invest, you just have to spend money, okay, to invest, okay, and then you get a payoff later, okay. So it's nothing comes for free. So that's what basically how the glycolysis works. So there's energy investment, which means they're actually using ATP. Okay, so during the energy investment, okay, they're utilizing ATP for the process. Okay, and then energy payoff is what, guys? That's when they get paid. Okay, which means like they gain more ATP during the energy payoff. Okay, so let's just take a look at, guys, energy investment. Okay, so glucose goes in, they start to phosphorylate glucose. Okay, which means what's a phosphorylation, guys? They use ATP and what, guys? Okay, so they put a phosphate group on them and ATP turns into ADP. So they actually use ATP for this. So again, what's the enzyme that does it? What's the enzyme that what? Takes the phosphate group and what? Puts it onto the guy like this? It's what? It's called kinase, exactly, right? So this is the first enzyme, and not that you have to know it, guys, it's what? Hexokinase. So something like kinase is what, guys? Their job is what? Take the phosphate group and put it onto the guy. That's what their job is, right? So basically what? Now you have what? phosphoglucose now. This is the first step of glycolysis, okay? Which means they're going to do like, okay, so they're using ATP for this, okay? And then like, okay, so not that it's important, guys. They convert the glucose into a fructose, phosphofructose, and then they use one more ATP, okay? So they use 2 ATP, okay, to turn into. So basically, what happens is they turn into the biphosphate phosphofructose. So which means what I'm saying is, guys, okay, one glucose goes in, what happens, guys? They utilize 2 ATP. 2 ATP has been expended, like they use 2 ATP, okay? So what is the, so during the energy investment phase, what, like, you know, this after 5th step, what do you get? You get something we call G3P. It's very important to understand what G3P is. I mean, so we might have talked about it last week, I'm not sure, guys. G3P is what, guys? Glyceraldehyde 3 phosphate. You got to know this for the AP. This is something that you might have to know for the AP. So G3P, you know, back in like old times when I was in high school, guys, they used to call this like PGAL. Okay, just so you know. So G3P is what? Glyceraldehyde. Three phosphate. Okay. So what the hell is G3P, guys, again? Glyceraldehyde 3 phosphate is what? It's a final product of what? Product of photosynthesis, Calvin cycle, okay? So there are a lot of enzymes, a lot of these intermediates, like, you know, these are called intermediates, right? A lot of these things kind of resemble what you see in the Calvin cycle, but it's kind of going backward, okay? So, and then like, yeah, like if plants produce G3P, what happens, guys? They just have to backtrack where they came from to produce what? Glucose, okay? So, you know, that's what they're doing, guys. So this is the, so there are 2 of these guys, by the way. So, you know, they split up. So basically, guys, again, so the term glycolysis is what? Split up the glucose molecule, okay? And the splitting happens in the what? Energy investment phase, okay? Because what? Glyceraldehyde 3 phosphate, this is a 3-carbon molecule. So the glucose is 6 carbons, right? So this splits up, and what happens, guys? You get 2 of these guys, okay? So that term glycolysis actually occurs in the energy investment phase of the glycolysis. Do you guys really know this part yet? I don't think so. You guys, you know, but you might want to know G3P. Okay? And then what happens? So I mean, they use ATP instead of gaining ATP. Okay, so what do they do, guys? There's an energy payoff. So energy payoff of the glycolysis process, what, guys? Yeah, so G3P. So basically, guys, I mean, G3P that you got converts into something you probably heard before, right? The pyruvate. Okay. So you got 2 G3P, now you got two pyruvates, okay, which is 3-carbon molecule. Okay. So during this process, okay, G3P to pyruvate, it produces, this is also a 5-step process, okay? We don't have to know the steps, it's fine. You produce 4 ATPs, okay, using kinase, okay? It's all by kinase, okay? And also what, guys? It also produces what they call 2-NADH. Okay, we have to talk about that, guys, okay? What the hell does it mean by this? So you gotta understand, guys. So they say what, guys? Overall process glycolysis, what happens, guys? By the net amount, you get 2 ATP, and also what, guys? 2-NADH. Okay, and that's what's happening. So let's take a look at, guys, okay? So I mean, ATP is like, okay, that's what they got, okay? They use kinase, the enzyme to get the ATP, right? Like, yeah, like they take it away, energy comes out, blah, blah, blah. So ATP comes out. The steps are not important here, but you gotta understand yourself, guys, okay? So what about this guy? NADH, what the hell is happening there? So keep in mind, guys, okay? Look at this. This is an aldehyde. So by no means, guys, I'm here to teach you guys like organic chem here. This is an aldehyde, okay? Pyruvate, by the way, okay, not a different name, but they also call this what, guys? Pyruvic acid. They also call this pyruvic acid, okay? So you're turning an aldehyde into a pyruvic acid, okay? So, dude, like this only time that someone will mention, your teacher will not say this at all. You'll never hear this, guys. I mean, until you go to college, okay? So when you are turning alcohol, okay, into aldehyde, okay, aldehyde into an acid, okay? So alcohol to an aldehyde to an acid, okay? This process, I mean, so again, like I'm not gonna go crazy right now, guys. This process is what, guys? Is what we call the oxidation process. Which means, well, oxidation is what, guys? You're removing electron. Here's what I'm saying right here. So if you're turning an aldehyde, which is G3P, glyceraldehyde 3-phosphate, into a pyruvic acid, here, what's going on, guys? You're what? You're taking the electron away from G3P. You know what I'm saying right here? So, and using the energy of that, blah, 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 you know, they're turning into a pyruvate. So, which means, guys, an aldehyde, which is G3P, to pyruvic acid. This is what, guys? You're removing the electron. That's an oxidation process, okay? You gotta understand, man, okay, just so you know. In bio, they like to talk about a coupled reaction a lot. Coupled reaction, right? What's a coupled reaction, guys? Yeah, dude, okay, you take, like, the reaction occurs, and there's a reaction that happens at the same time. That's a coupled reaction. Without one or the other, you know, reaction's not gonna happen, right? So G3P to pyruvic acid, or pyruvate, okay? So this is an oxidation, which means what, guys? There must be a reduction reaction happening at the same time, okay? This is what? It's a coupled reaction. So oxidation must be coupled with the reduction process. So what's the reduction process? This is it. This is important, guys. Where NAD plus, okay? So this is like nicotinamide, like dinucleotide or something. That's what they call it. Okay? It's not that you have to know what it is. The naming of it's fine. NAD plus 2 electrons. Okay, the electron is what? Okay, so reduction. They're gaining electron, right? Plus H plus turns into NADH. So you gotta understand, guys, by the way, okay, so in order to, so what is really needed for glycolysis? You need what, guys? You need to have a glucose, you need to have what, guys? NAD plus. Because why? As they run the oxidation for energy payoff, what happens, guys? They need to put in NAD plus in here. So you couple with what? Oxidation of G3P. Okay, so that's where this guy comes from, NADH. Okay. So any questions on the rest for this glycolysis? I mean, like, dude, like it's not too bad. I mean, it's you just have to know what's happening. Any questions on the rest? So this can be done by what, guys? Like every cell that you know. Okay, like dude, like even for prokaryotic cells, they're doing this. So you know.
Just 2E minus plus what?
H plus.
H plus.
Yeah, H plus.
And all this takes place in the cytoplasm.
Correct. Yeah, you can think about it, right? Like prokaryotic cells, they don't have a membrane-bound organelles, right? Which means, okay, so yeah, this must happen in cytoplasm, right? So like you gotta understand, right? The enzymes, like such as hexokinase, you know, like there's other kinase here, you know. So these enzymes are what, guys? They have to be what produced, like we talked about, right? The ribosome, right? And cytoplasm ribosome, okay? And then yeah, like not the the rough ER ribosome, right? Because what, they're being utilized in the cytoplasm, like blah blah blah, okay? So yeah, if you are a cell, okay, like if you think about it, guys, if you are a prokaryotic cell, okay, which like this is it, like dude, the 2 ATP they create from one glucose molecule, dude, that's it. That's it. That's all. That's only way to produce ATP for them. Okay, 1 glucose, 2 ATP, and other stuff is what, waste. You know what I'm saying? So we understand, like you probably know, guys, okay, oh yeah, I know there's extra step to this, which is more ATP, but that's for the eukaryotic cells only, right? But now, yeah, what is a problem for the prokaryotic cells right now? So yeah, they produce, okay, we understand they got ATP, which is what they needed. Dude, that's it, right? Okay, but keep in mind, okay?
Byproduct.
Huh?
The byproduct.
Yeah, like, okay, so byproducts, NADH is a byproduct. So what's a byproduct, guys? Something they produce, but they don't really need, okay? So they produce NADH and pyruvate. Okay, they can throw this out. They can throw the pyruvate out. Oh yeah, bye bye. You know, like we don't need you anymore. Okay, bye bye. You know. But can they run this? So another glucose comes in. Okay, can they run this process all over again? So yeah, they got ATP. So out of 4, I'm gonna put 2 ATP again in here, and then I'm gonna get 4 again. Okay. So at what? If the plenty of glucose is available. So the question becomes, guys, because if you just have glucose, can you run this process all over again? Can you run this like every time? What do you think? Can you? It's like, yeah, throw all these out. Okay, I'm gonna run the glucose again and then run this. I mean, so enzymes are okay. Enzymes are okay because why? Enzymes never really get, like they don't get used up, right? What's the problem with that process right now? What do you think? Why can't they just like, oh yeah, I'm gonna throw everything out because ATP is what I need, okay? And then, okay, so I'm gonna have another glucose come in. So what's the problem with this? With these prokaryote cells, what do you think? Yeah, what do you think, Andy?
NAD plus.
Exactly, yeah, the problem is this guy. You know what I'm saying, guys? Yeah, so they can't just produce they can't just produce this like, oh yeah, I'm gonna produce one more of these guys. They can't do that, okay? So keep in mind, okay? They have to do this process again. What is the problem, guys? They need this. They need to what? Convert NADH into what? NAD plus. So truly, guys, okay, what really bottlenecks the process is what, guys? They gotta get this back. So which means what? Okay, they produce NADH. Okay, they have to convert this what? Convert this back into NAD plus. Okay, because why? The reaction is coupled. So now the question becomes, how the hell do they do that? Yeah, so what do they do, guys? So if you are a prokaryotic cell who, like, with the steps like, glycolysis, their only way to get ATP, what happens, guys? What do? They run something they call what, guys? Fermentation. They do something called fermentation, right? So, dude, like, you gotta understand, guys, okay? What's fermentation? So fermentation may seem, guys, like it's not needed step, but it's very crucial, okay? Fermentation, what, guys? Okay, so they turn pyruvic acid, okay, or pyruvate. Okay, I'm not gonna tell. I mean, so what's the difference between pyruvic acid and pyruvate, guys? So what's the difference, guys? This is actually pyruvate. You don't have to know the shape, guys, but it's fine. This is a pyruvic acid. Yeah, what is a pyruvate? What do you think? APChem? What do you think, Andy? Acetic acid, acetate? What do you think?
Get rid of the O.
Exactly. Oh, wait.
The O.
I don't know. Yeah. That's what? That's a pyruvate. H, like acid, H plus pops out. So acetate, acetic acid, okay? So H plus given off. It's an acid. Because you know what I'm saying, guys? Okay? So this is a pyruvate, okay? This is, when the H is there, that's a pyruvic acid. Okay, so that's the difference. So either or, it's fine. Okay, so you can say pyruvic acid or you can call it pyruvate. It's a weak acid, so you can think about it because how much of an H plus have dissociated, or came out, like that's the question you have to ask yourself, right? That's a pyruvate, okay? So what do they do with this guy, pyruvic acid, in the fermentation? So you probably heard, right? Alcohol fermentation. What do, guys? They convert this into what, guys? Ethyl alcohol. Acid into an alcohol. You're turning an acid back into an alcohol. What is that, guys? That is a reduction process. You're going backward. You know what I'm saying, guys? So again, yeah, alcohol fermentation is what? Okay, so you're turning into an alcohol, which is 2 carbon and what? Plus a CO2. Okay? This is what, guys? A reduction process. Okay? Again, this is fermentation, right? Fermentation, whatever. Okay? So if you're trying to run a fermentation, which is a reduction process, okay? Yeah, what do you have to do? You must have a what? You must have an oxidation reaction, right? And what is the oxidation reaction, guys? Okay, so I have NADH. I'm gonna make this into what, guys? NAD plus 2 electrons plus H plus. So that's an oxidation. Okay, so by doing fermentation, what are you doing, guys? Okay, so I'm what? You're turning it back into NAD plus. Why? to replenish the NADH to run for the next glycolysis process. Okay, so the fermentation process is not something like it's an extra step for them to what? Not produce ATP at all. It's like, and this is the alcohol that we utilize to what? Make make bread and like the drinking alcohol. You know, not for you guys right now, but you know, but why do they do this, guys? Because what? Yeah, return NADH into what? NAD plus. Okay, that's the main purpose of this. Okay, so again, there are two types of fermentation, guys. You have to know, which is what? Lactic acid fermentation. Lactic acid fermentation and what? Alcohol fermentation. There are two. Okay, so you know, a lot of times, guys, prokaryotic cells produce alcohol they do alcohol fermentation, and you probably heard, guys, your muscle cells will do lactic acid fermentation. Okay, so you know, so that's at an anaerobic perspective, and you should understand. I tell you, this is part of the AP. They will ask you guys, what's the purpose of fermentation? Okay, why the hell do they do that? And your answer is, look, guys, okay, turn this guy back into an NAD plus to utilize them for what? Next glycolysis energy payoff. Okay, that's your answer. Oh my God, this kid knows something. That's what it is. Okay. Any questions, concerns? Any questions, concerns?
So what's the purpose of the diphosphofructose? What is it?
Diphosphofructose. So if they ask you what the hell is diphosphofructose, my answer to you is that's intermediate of the glycolysis. So remember I told you there are 10 steps. So they first convert the glucose into a phosphoglucose, turns to phosphofructose, biphosphofructose, and then turn it into G3P. Okay, so it's a step. And then so they go into energy payoff now, which they turn into G3 turns into phosphoglycerate, like biglycerate, and then phosphoenol pyruvate turns to pyruvate. So if you tell me, okay, what the hell is the biphosphofructose? My answer is it's a step. It's something that was produced in the steps of glycolysis, which you can call what? As what they call intermediate of the glycolysis. That's what it is. So not that you have to know all of them. It's fine. I mean, dude, like AP, like I'm not concerned. Okay, they will never ask you. Oh yeah, by the way, you know what? No, they'll never ask you. They only ask you what goes in, what comes out, what happened. That's what they ask you. Any questions, anyways? So, is there any more, guys, that you guys have to know other than this? Of course, yeah. Right, like, so, our cells have what, guys? Our cells have, you know, more stuff, right? We have mitochondria. The mitochondria can produce more ATP. So, like, dude, we got only 2 ATPs. That's it. So, yeah, look, think about it, right? So, if you have a little chem background, because there's a lot of stuff we're discussing, like chem-related stuff, right? So, let's take a look. So, glucose molecule, C6H6. So a lot of you guys did chem last year, right? You know, you guys have to understand, guys, the oxidation state, right? That's one of the stuff you did, right? Hydrogen is charged at +1, oxygen is charged at -2, okay? And then you can derive what the charge of carbon is, right? So +1, they're 12, right? So you have +12 charge. And this is what, guys? Like they're -2, 6 of them -12. So the whole thing does not have any charge at all, which means the carbon should have charge as 0. Do you understand what I'm saying? Okay? So carbon, dude, like I'm not trying to question you right now, guys. Carbons, right? If there was 0 charge, what does it mean, guys? They did not lose or gain electrons. That's what it means. Okay, which means what, guys? They still have what? Their 4 valence electrons. You know what I'm saying, guys? They didn't lose it. They didn't take more. They didn't lose more. Okay, which means what? There are 6 carbons that has they're 4 electrons each, which means what? They have what? Overall, they have what? 24 electrons. Okay, they have to take out. You know what I'm saying? So, yeah, during glycolysis process, okay, yeah, so look at this. They produced 2 of these guys, which means what? What does it mean? They only extracted 4 electrons out of 24 from carbon. So the carbon, 6 carbons have what? 24 electrons. They only took out 4 of them, which means you got 20 electrons left. You understand what I'm saying? Okay. So it's such a wasteful to get rid of the 20 electrons. What do they do, guys? We have mitochondria, which is what? Specialized cell, okay, that will what? Extract all of this, okay? So there's extra step, right? What the hell do they have, guys? They have a steps of what, guys? Krebs cycle. So so the Krebs cycle, right? All you have to know for the Krebs cycle is what? What do they do? Extract everything out. Take all the electrons out of the carbon, okay, and produce CO2. That's what the Krebs cycle process is. Let's take a look, guys. So, so guys, I mean, so keep in mind, guys, before I move on, guys, okay, the glycolysis, the ATP production. So you got to know this, guys. The ATP production in glycolysis, they call this, okay, substrate level. Substrate level phosphorylation. Okay. You gotta write this down, okay? It's called substrate level phosphorylation. So production of ATP during glycolysis, okay? They call that, so phosphorylation is what, guys? Phosphorylation is where they put the phosphate group on them, right? It turns into ATP, right? So the phosphorylation is what? Like, yeah, they call it substrate level. It means what? They're using an enzyme, something we call kinase. Okay. And what? Yeah, like, okay, so they take, it's enzyme catalyzed phosphorylation. Okay, that's called a substrate level phosphorylation. Okay. So guys, I mean, so yeah, they produce 2 ATP by the net amount, and then let's take a look at what happens after this. So after this, guys, what will happen is, they call this process Krebs cycle. So this is mitochondria. Again, my drawing, guys, is not the best, you guys know. So mitochondria has an outer membrane, okay, like the phospholipid, and the inner membrane. It doesn't look like this, okay. You know, you can look at the textbook for the video, but I can't draw that well, okay. So yeah, one of the questions on the homework, right? If you have to take a look at it, okay. The question was, okay, how does the having a double membrane? So like, dude, they have an outer membrane and inner membrane for the organelle. Okay, there are no other organelle that has this. Okay, except for chloroplast and mitochondria. And the question was asking, okay, like I remember there was a question, epicoplast or something on the homework, right? So, you know, and the question is asking, okay, how do you know having a double membrane like this, okay, help you to understand that mitochondria was once a cell of its own, okay, but was engulfed by the larger cell. So what was like, you know, like I saw some of the response that you wrote, like if you've done your homework like much earlier, is what, guys? I mean, so again, if a smaller cell gets engulfed by the larger cell, what happens, guys? I mean, so they create them into what? A vesicle. Okay, so like think about it, guys. This is a larger cell, okay, and this is a smaller, tiny, you know, cell. Okay, that has a membrane. Okay, like at least a cell has some membrane, right? If this cell is engulfed, okay, that's endosymbiosis understanding, right? And what happens? Okay, so which means, okay, so they're gonna pinch in like this, you know, and then like this guy is gonna come in like this, and what later, guys? What happens? It will turn into a vesicle like this, right? That's what happens, right? So why do they have a double membrane? Yeah, like yeah, look at this. Because why? They came in by the method of endocytosis. Okay, that's why they have a two membrane like this. You understand what I'm saying? So okay, so that's one of the understanding of a dual membrane, double membrane as well. One of the understanding of what? Oh yeah, because what? They probably, no one knows because it happens probably billions of years ago. Is what? Yeah, that's what happened. Like, as I said, right? The first, they suspect the first appearance of the plant on land was 500 million years ago. Okay, I said this, right? And the first appearance of the fish coming into land is 350 million years ago. Okay, yeah. When do you think this happened? What do you think? Probably what, guys? Like billion years ago, maybe? So can anyone like really know what happened? Absolutely not. They all say this what? They suspect. They can't be for sure. Like, oh yeah, this is what happened. They can't say that. It's why? Like, how the hell do you know? I don't know what happened yesterday. I forget. How do you know what happened a billion years ago? So they only what? Speculate. Okay? Yeah, it could be. That's what happened, right? So, you know. So that's why they have a double membrane, right? So this is what they call this space between called intermembrane system. Okay, I'm not gonna read it. Intermembrane system. It's like intermembrane between the membrane, right? Inside here, okay, it's called matrix. So basically what happens is, guys, the pyruvate they produce, or pyruvic acid, whatever, they go into the matrix. Okay, this is mitochondria. Okay, they go into a matrix. So before they enter the Krebs cycle, you gotta know this all, guys. Like this is all AP. Okay, if I think you don't know it, guys, I'll say you don't know it. Okay, you don't have to know it for the AP. So now, before they get into a Krebs cycle, which they call this a citric acid cycle, there's like some step to this. So a pyruvate, this happens in the matrix. So AP guys, what do you think they'll ask you guys? They'll ask you where the glycolysis occur. I mean, yeah, cytoplasm, right? And then they'll ask you where does the next step occur? Okay, matrix, of mitochondria. Okay. So, and like you can, I mean, I never seen them ask guys how do they go in. Okay, I never seen them ask that question, by the way. Okay, but again, like whatever. So, pyruvate. So before they enter a Krebs cycle, a citric acid cycle, okay, there is an extra step prior to going in. So, I think like what did they say? Like one of the students told me their teacher mentioned this as a linked reaction or something. I don't know, link reaction. Okay. I never heard this term, linked reaction. I don't know. So what was that? Who said that? I mean, one of the students from one of the high schools nearby. I don't know. It's not Jericho. It's like Whitley or something. I don't know. But I never heard this term. Okay, linked reaction. I don't know. Maybe links the Krebs cycle and the glycolysis. Maybe I'm not sure. But pyruvate, okay, what they do is what? They turn the pyruvate into, I'm sure you heard this, like, okay, acetyl-CoA. You have to at least know this process, okay? Acetyl-CoA. They turn pyruvic acid or pyruvate into what? Acetyl-CoA, okay? And during this process, what happens, guys? They produce CO2, okay? And what produce what? Another NADH. 2 NADHs, okay? So pyruvate turns into what, guys? Acetyl-CoA. Okay. Keep in mind, guys. Pyruvate is what? Pyruvate is 3-carbon molecule. CO2 goes out. What happens, guys? This is what? A 2-carbon molecule. Acetyl-CoA. Okay. So this is what they consider as a linked reaction. Okay, which happens in mitochondria. So, and there's nothing more I want to mention about this one. Okay. And then what happens, guys? In the matrix. That's why they start. So with this guy, acetyl-CoA, now they will run the Krebs cycle in the matrix. So what happens is now the Krebs cycle. It's a cycle, okay? They call this a Krebs cycle. The guy who figured this out, his name is Krebs, I think. And then, or sometimes they call this guy citric acid cycle. Citric acid cycle. So you don't have to know the steps here, okay? Except for some folks here, okay? So what happens is the acetyl-CoA goes in, okay? Acetyl-CoA that was produced. Acetyl-CoA. So, okay, dude, like, unless your teacher is like really about this, okay? You don't have to know how many NADH get produced. I don't know, okay? So there are 2 pyruvates, right? Because we split up, right? 2 pyruvates available, and probably they have what? Overall 4 NADH popped out because there are 2 pyruvates, right? Because why? One glucose went in, is split up. Okay, now we have 2 pyruvates, right? So if you think about it, right? So what happened is overall you took out what? There are 10 NADHs that has been produced since glycolysis. Okay, glycolysis produced 2. I'm sorry. No, 6 NADHs. Sorry, 6 NADHs. So if you think about it, guys, I mean, so 2 NADH from glycolysis, okay? 4 NADH from the linked reaction, okay? The total of what produced 6 NADH. So how many can you get, okay? Like overall process-wise, from glucose molecule. So if you think about it, right? Again, glucose has 24 electrons overall, okay? Which is what? That is what? 12 NADHs. Right? So each NADH you need to put 2 electrons in there. So okay, you got 6, which is what? You're only halfway done. So the Krebs cycle is gonna do what? Everything else. Okay, they're gonna take out what? Every electron that's out there, which is what? Yeah, they're gonna produce what? Close to like 6 NADHs. That's the job of this cycle. So acetyl-CoA. So there's it's a cycle, right? So why? So there's a thing called oxaloacetate. You don't have to know this, but it's fine. Unless your teacher is crazy, guys, it's called the oxaloacetate. Okay, will bind to acetyl-CoA and produce something called citric acid. Okay, it's a part of the cycle. So oxaloacetate in the matrix, acetyl-CoA, okay, they will react and turns into what, guys? Citric acid. Okay, and so that is the reason why they call this what, guys? A citric acid cycle. Because you know what I'm saying, guys? It's first product. So the other stuff, guys, here, I don't think you need it for you guys. I don't think you have to know any one of these guys in your steps. Okay, so they call like isocitrate. You know, they produce succinate, succinyl-CoA, malate, you know, and then oxaloacetate. Like, you don't have to know all that. Okay, for AP, I don't think. I mean, you can take a look at your reading, guys. If I haven't provided you guys later. So you don't have to know any of these steps, okay? So you know, all you have to know is what, guys? They produce remaining NADHs. Okay, so they produce here. If I'm not mistaken, guys, they produce 4 additional NADHs. And I think they produce 2 FADH2. Okay. So which means, guys, I mean, yeah, FADH2. So there's a different NAD. I mean, so different, you know, electron carrier like this NADHs. They call FADH2s. Okay. So that's basically what occurs in the Krebs cycle. Okay. And it's a cycle where this is a malate converts into oxaloacetate. Okay. And during the process, what happens, guys? They produce what? They produce 4 CO2s. All the CO2s out the door. So carbon all turns into a CO2. That's what citric acid is. Okay. Which means what? They what? Oxidize every carbon that's out there and converts into a CO2. So, yeah. Ask yourself, guys. Okay. You're breathing out CO2, right? Yeah, dude, like, okay, so if you run for a long time, or even right now, you guys breathe out what? Carbon dioxide. Okay? Our cells produce CO2. You produce CO2, right? Like, you know, where does it come from? Yeah, where does it come from, guys? Yeah, dude. The linked reaction produces CO2, 2 CO2s, okay? And also what, guys? The Krebs cycle produces CO2, okay? So, you know, detail down the road, okay? Like down the road, if you think about, okay, so where is my CO2 coming from my body? Is well, oh yeah, it's coming from your cells, you know, mitochondria, okay? Where they what? Run the Krebs cycle, they produce CO2. That's where the CO2 comes from your body, okay?
So can you say the order again? So we start with acetyl-CoA and then we do citric acid and then keep going like that.
Yeah, like it's a cycle, right? They turn it into isocitrate. And then, like, I forgot some of the T side. I forgot to send it. So succinyl-CoA, I forgot, there's one here. And it's succinate, and I think they turn into malate, and then into oxaloacetate. There's 8 steps here. So, you know, you don't have to know any of this.
What's that on the left? FADH?
FADH2. So it's FADH2. It's good to read. So this process also produces 2 ATP as well. Okay. 2 ATP overall, 2 cycles. Okay, like there are two acetyl-CoAs, right? 2 ATPs. Okay, so again, this is also what, guys? Based on what? How do they produce ATP, guys? Is what? Based on the substrate level phosphorylation. Okay, you see what I'm saying, guys? Alright, so you got what I'm saying, guys. So basically, they produced a lot of these NADHs, got FADH2s, and maybe 4 ATP, and what now? So they only produce 4 ATPs now. So what do they do now? They've got these NADHs. What are they going to do with this? Is the question to ask yourself. Okay, what are they going to do with NADHs? So next is what they call, guys. They asked this a lot of AP, by the way. not like these things, but they ask a lot about this now. And you have to know exactly what to write on them. So this is, now guys, something we call electron transport chain. Remember, electron transport chain. You gotta know this, ETC, electron transport chain. So, take a look at it, guys. So all the NAD, this is matrix, okay? This is an intermembrane space, okay? Intermembrane spacing, okay? So this is outer membrane. So this is a between. This is a inner membrane, sometimes called the crystal membrane. Intermembrane matrix, okay? We have produced all these NADHs, NADH, and also FADH2. So what the hell do we do with this? So let me explain to you guys you know, like what's really happening, because if you read the textbook, guys, that's what they say. So this has to do with, guys, the energy of electron, like, you know, energy of electron overall during the overall process. So this is the energy of electron when electron is bound to a carbon. Okay, when the carbon is an electron is bound to a carbon, that's where you're at. Okay, it's a relatively high energy here. Okay, and now keep in mind, guys, here the electron will be accepted by later by the O2 gases. Okay, so when the O2 gas binds to electron, that's the energy of the oxygen. So since you guys have a little background in chem, okay, so now, so keep in mind, guys, by the way, so you guys understand, guys, like the electron, like principal energy level, guys, from AP Chem, right? So keep in mind, guys, okay, this is like, for example, guys, N1, okay, this is N2, N3, N4, like N5, and this is N like 6 or something, okay? So, like, again, electron wants to come down to a lower level, right? This is one of the stuff you should know, right? Because why? The nucleus has a positive charged proton, right? They want to come down. Why? Because what? Dude, like when they come down, what happens, guys? The potential energy of electron lowers, okay? They want to come down because this is unstable, high energy. This is what? Low energy. They want to come down, okay? Keep in mind, guys, okay? Carbon has much, this is AP, okay? Carbon has much weaker or lower the term electronegativity, okay? They have less attraction for the electron. So when electron is bound to a carbon, is what? They're more loosely bound. They're like further away, okay? So, which means what? If the electron is more further away from the nucleus, what happens, guys? Yeah, they're weaker. I mean, so, which means what? They're what? They have a greater potential energy, loosely bound, okay? It's easier to take them away. So, if you put the electron to the oxygen where it has a very strong affinity for the electron. So, oxygen, like what? It attracts electron, right? Which means, like, yeah, dude, they're losing electron. Losing what? Electron is losing what? Energy. They're very freed up. Like if they're freed up, they can do whatever they want. And what happens, guys? Oxygen is like tightly holding onto them. What happens, guys? Yeah, the energy is lost. Okay, that's what's happening. So basically, what's happening is what, guys? Electron is what? Captured now by what? NADH. So they use some energy to what? It comes down to NADH. Okay, and also what? It comes down to what? FADH2. So basically, what's happening is what, guys? The high energy electron is what, guys? Yeah, dude, it's being carried by NADH and FADH2s, okay? So, you know, but they have what? This much energy still available to do work, okay? So, you know, so basically, you can take a look at it, guys, as what? NADH is what? Electron carrier, okay? For the last step that we have, which is ETC. And how are they going to utilize this much energy, okay, to produce more ATP, right? So still at the moment, guys, they have energy stored in the NADH and FADH2s, and what are they gonna do here? So, so what happens is, so these, this is actually got intermembrane space, okay? They got some of the proteins on it, okay? You don't have to know any one of these guys' proteins, by the way, for the you don't have to know, guys, any one of these proteins at all, okay? For you guys, for AP. They will not, it's not in scope for you guys. So, these are cytochromes. cytochrome, okay, is this flavor protein, FMN. So this is called, one of the protein there, guys, is a ubiquinone. You don't have to know them, guys. If they call it U here, and this is FAD. Cytochrome. This is the ATP synthase. You have to know this guy. You have to know this guy, ATP synthase. Okay, but you don't. Maybe the term cytochrome, maybe. Okay, cytochrome. FMN, no. Okay, ubiquinone, no. FAD, no. Okay, you don't have to know any of those. Absolutely none of those. Okay, except for like some of the folks here. Okay, you guys have to know them. So what they're doing is this. Okay, basically what they're doing is they're using NADH and FADH2s. Okay, and what the electron is what given to. So NADH, they're giving the electron away to FMN. Okay. So electrons, so NADH, it turns into NAD plus H plus 2 electrons. Electrons given to what? FMN. So remember, guys, what we talked about? Yeah, to run. So keep in mind, guys. Okay, this is what they call aerobic respiration. Okay, to run this process. Okay, again, to run this process, they need what? They need to run glycolysis. Okay, and now. What was really important about the glycolysis process, guys? What? Replenishing NAD plus. You understand what I'm saying, guys? They gotta get the NAD plus back. Okay? This is the way to get it back. Okay? Which is what NADH is what? Giving away the electron to FMN. Okay? Which is a flavor protein. So they're handing away, okay, the FMN, electron to FMN. So like NADH is what? They lost this much energy. Okay? Like here to here, they lost energy. What are they gonna do with the energy, guys? So you had like 100 of energy, you lost to 50. What the hell is gonna happen, guys? FMN will be using this energy, okay, difference of the energy what? To, listen carefully, guys, actively transport H plus from the matrix to the intermembrane spacing. Okay? This is active transport. They're using the energy, okay, from the matrix to intermembrane space. So here's what I'm saying. So from NADH to this, they're using the energy to pump out the H pluses. You guys understand what I'm saying, guys, here? And then what happens, guys? And then the electron. So electron lost energy, right? Like electron lost energy. What happens, guys? It will be handed down to the next one, which is the next cytochrome. So the electron will be following this way and what? Handing down to cytochrome. Cytochrome uses what? Extra energy as well to what? To pump out more H pluses. Okay? And the electron will be what? Will be going down to the another cytochrome and what? Pump out H pluses again. Okay? So ultimately what happens, guys? Yeah, they'll be handing down and what? Go down to what? It goes down to, if you look at it, guys, okay, so this is the cytochrome 1, cytochrome 2, and what? They use the energy to pump out these pluses. Okay, so now there's what? Much greater H plus concentration on what? Intermembrane spacing. That's what they do. Okay, that's basically what they utilize the energy for. So, so all in all right now, guys, okay? So what they're doing is what, guys? Okay, they use the high energy electron, okay? This is mitochondria, right? This is the inner shell matrix. That's the intermembrane spacing. So what do they do, guys? They're pumping out these H pluses into the intermembrane spacing. Okay, that's what they're doing. There's greater concentration of H plus. So one thing to note, guys, okay? So the electron is here. Yeah, what must happen to the electron, guys? Electron must what? So think about it, guys. There are electrons lined up. It's like what, guys? It's a chain reaction, right? Yeah. One electron went in. Okay, they pump out H pluses, right? Yeah. What must happen? The next electron must go in. It's like a line. Okay. So this electron must come out. Okay, they must freaking come out, right? What happens, guys? There's what? O2 gas takes the electron away and what? Turns into H2O. So yeah, you guys breathe in O2 gases. What do they do, guys? In the electron transfer chain, this is what the O2 is used for. This is what? They call this what? The last electron acceptor. It's called the last electron acceptor for what? Electron transfer chain, okay? So it comes down to the O2, right there, okay? Yeah, look at this. So you gotta ask yourself, right? I mean, like before we talk about anything else. So without the O2 gas, so you stop breathing in the O2 gas, okay? Like, not you, because someone else, guys, underwater, whatever, okay? They stop bringing in the O2 gases, okay? Like people can hold like the whatever, okay? Breath for like a minute or two. I don't know, okay? How long you can hold a breath? I'm not sure. But yeah, think about it, guys. If O2 gas is not available, okay? Like you probably heard this, right? What do? If your body muscle, okay, body cells do not have the access to O2 gases, what happens, guys? Well, yeah.
Fermentation.
Because yeah, fermentation. Your cells switched off to what, guys? Fermentation. Yeah, why? Where? Why?
It doesn't need oxygen.
Huh?
It doesn't need oxygen.
I mean, but I understand. Fermentation doesn't. But why do they switch off to fermentation? That's my question.
To make ATP.
Yeah, but why? Like why? Like I understand they want to
We
make.
can't do this process.
Why?
They don't have oxygen.
Huh?
Because
So you
it's
gotta understand what I said, right? Like, so the reason why they switch over to fermentation is what? The main thing, like keep in mind, the main thing that really stops the entire process, what? Lack of NAD+. Okay? So the electron transfer chain, what does it do, guys? Having the O2 available is what, guys? They replenish NAD+. Okay? So without the oxygen, what happens, guys? NAD, this process will not occur, which means what? Everything is held up by what? Held up as NADH. Okay? If you do not have NAD+, what happens, guys? You are not able to run glycolysis. You know what I'm saying, guys? Glycolysis will not occur. Because so, like again, glycolysis is what? It's oxidation process. It requires what? Reduction process of NAD+ to NADH. We talked about this, okay? So without this, what happens? They can't even run glycolysis, okay? So what do you do, guys? Your body switched over to what? Fermentation to what? Turn this guy back into what? NAD+ by running fermentation. So truly, okay, so we understand, okay, your body, without the availability of O2 gases, is switched over to what? Like lactic acid fermentation. But you got to ask why? Because what? They're converting this guy back into what? NAD+ using fermentation. That's why they're doing that. You know, so it's not because they need more ATP, yes, okay, but to run the basic steps of glycolysis, they need NAD+. Because they're held up here. What happens, guys? They're running this into what? They're running fermentation, okay, and replenish back into NAD+, at least one glycolysis, okay. So you gotta understand, guys. Okay, I mean, again, dude, I tell you guys, your teacher will not talk about, oh yeah, I just know that. Okay, that's it. That's the next chapter. Dude, a little more than that, okay? A little more than that, okay? So, you know, so like you gotta understand, like why, like what is the main thing that makes them switch over is what? To convert NADH into NAD+. Okay?
So you need oxygen to do that.
What's up?
You need oxygen to convert NAD+ to NADH.
No, fermentation does not require oxygen gas at all. Fermentation basically is what? Convert the pyruvic acid into pyruvic acid. Pyruvate or pyruvic acid into what? Lactic acid or alcohol fermentation. So that process, reduction process we talked about, which will turn NADH into NAD+. Okay, that doesn't require, because the oxygen is not there, they're switching over to produce NAD+, which is utilized, again, for the energy payoff during the glycolysis to produce more ATP. So I mean, like, whatever, like, we have to go to that, that's fine, that's fine. But like, oh yeah, the oxygen gas is not available, then yeah, like you switch fermentation. Will they ask you why is the question, right? They will not ask you why. AP guys, no, actually no. So again, what will happen is, so one more thing, guys, right? Cytochromes. Cytochrome, okay? Put your phone away. Okay, cytochrome, guys, okay? These cytochromes are, again, a lot of the cells have cytochrome, okay? Like meaning, guys, if you run electron transfer chain, they got cytochromes. So you know, which means, guys, this is also a very conserved process, okay? Like, so what I'm saying is, guys, this, okay? like if they run evolution, right? Evolution again, like evolution is a big topic, guys, in bio, by the way. Okay, so what's the best way to tell, for example, guys, how close a human is compared to like chimpanzee or whatever? What do they do, guys? DNA sequencing. Okay, DNA sequence, right? Like, yeah, compare DNAs, right? But now, what's the next best thing? Compare what? Amino acid sequencing. Okay, so which amino acid sequencing look at? Yeah, they have to look at a protein, right? So what protein they look at? Cytochrome. Because what? Every cell have what? This is a very conserved process, which means what? They use what? Cytochrome. Okay, to like, yeah, because everyone has it, right? So cytochrome is what? Typically cytochrome is an iron heme in there. It has iron in there, guys. Not that, I don't know if you have to know that, guys. So like hemoglobin, they have iron in there. So guys, we haven't really produced ATP at all. So we produced these concentration gradient of H plus there. There's more H plus in the intermembrane spacing, guys, but we haven't really produced ATP at all. So what happens is, because they have a lot of H plus build up, what happens, guys? They got something they call ATP synthase. It's called ATP synthase. Okay, that's the enzyme that they got over there. Okay, so what they'll ask you is about this guy. Okay, are you cold? I mean, I'm like hot, guys. But I mean, like the temperature-wise, I'm like sweating. But all right. So guys, what do they do? I mean, so how does that work? It's synthesis, guys. Yeah. So yeah, listen very carefully now. So there are some terminology you have to utilize. Okay. First terminology is what, guys? So the electron transport chain of pumping out the H pluses, which is a proton, right? What happens? It produces what? It produces, you can very carefully, electrochemical gradients. You gotta write this on your AP exam, okay? So it's called the electrochemical gradient of H plus. Okay? So we talked about this a little bit, guys. We call this electro because why? Because it's what? This is a charged particle which will create what, guys? Electric potential voltage. Because I don't want to go crazy with you guys right now. It's a voltage value. Okay? Chemical because why? There is more of H particles, hydrogen particles outside the membrane. Okay? So here. Right? So that's what they call electrochemical gradients. Okay? If this didn't have a charge at all, what happens? We call this what? We call this what? Just chemical gradients. Okay? So now, using the electron transfer chain, okay, now we produce what? Electrochemical gradient of what? Yeah, H plus. There's a higher concentration of H plus in the intermembrane spacing compared to the matrix. There's a lot more of that kind. Okay, so AP will ask you, oh yeah, like prove this. Like, prove what? Like, you know, like yeah, what's the indicator? AP, like, dude, AP is like, I don't know, they play with words, guys, by the way. So they'll ask you, yeah, how do you know H plus 1 there? Like, how do you measure this? How do you measure, okay, there's more H plus here? Yeah, what's the best way to measure Andy, what do you think?
pH?
Because yeah, okay, guys, pH should be higher, right? Andy? pH should be higher, right?
Lower.
Oh, okay. pH should be lower, exactly. Okay, so yeah, pH should be lower here compared to the matrix. Okay, so yeah, pH is around like 3. pH is around 3, guys, versus here. So one of the indications like pH high, or what? This will have what? Greater voltage, okay, on this intermembrane spacing. Greater voltage, greater electric potential value, because there's more positive charges, right? So that's an indication what? Oh yeah, this is happening, okay? This is going on. So electrochemical gradients, so they use the energy. So they use the energy of electron to what? Okay, it's produce the electrochemical gradient of H plus. Okay, and then what? Okay, so what the hell? And then what? So now ATP synthase will go to work. So basically, what happens is So again, there's a lot more to ATP synthase. The guy who figured out how this worked, he got a Nobel Prize for this. Okay, but you do not have to know very much in detail about this guy, okay, for AP. AP guys, oh yeah, like they don't care too much, okay? So when ATP synthase guys, basically what happens, they have an F0 unit, they have an F1 unit guys, by the way, and you will never hear the term F0, F1 unit guys of the ATP synthase. So basically what it does is, it brings in, so now, like, dude, what will happen is what? Charged particle, they will never pass the membrane. Okay, that's not gonna happen, right? They have to come through a channel, which is what? ATP synthase. So ATP synthase will open the channel. Like it's open the channel, guys. So F0 opens the channel, okay? And at F0, like the protein, like it turns like as turbine, okay? And then using the energy of the electrochemical gradient, they'll produce ATP. So they have a, dude, like you gotta understand, okay? When the H plus comes in, okay, they put a special term or the name for this. That is, you gotta understand, it's called chemiosmosis. So they use this term chemiosmosis, okay? When the H plus comes in through ATP synthesis, oh yeah, this what? They come in by what? Chemiosmosis. Okay? And using the energy of the electrochemical gradient, what happens, guys? They start to produce ATP, okay? So, and like, okay, so then what happens, guys? Use what? Yeah, ADP plus phosphate into ATP. So you do not have to explain how they do that. That's not part of the ATP will never ask you, yeah, explain how do they do this. Okay? There's alpha and beta subunits, okay? As they turn, it changes the F0 and F1, it changes the structure, and they will have enough energy. I mean, you'll never have to do that. That's not something for EP. Okay? So, and I think approximately one NADH will produce around 2.5 ATPs. One FADH2 produces around 1.5 ATP. Okay? So why NADH2 produces less? Because they start with this process. Okay? So, yeah, I don't know what the craze right now. So keep in mind, guys, way they produce ATP using this process, okay, listen carefully, it's called oxidative phosphorylation. You have to write this term, okay? Oxidative phosphorylation, okay? Which deals with what, guys? Producing electrochemical gradient of H plus, okay? And the H plus comes into the matrix backed by what? Chemiosmosis. ATP synthase produces ATP using the electrochemical gradient energy, okay? This entire process is called the oxidative phosphorylation. If you can utilize these terms, then you'll be okay with ATP, okay? You don't have to like cytochrome C, like blah, 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 like, so it's fine. F07 and F1, like, no, that's not something that you will need to explain to them. You just have to understand, guys. Okay, so these will pump out H pluses. I mean, it'd be nice if you know them, like FMN, like flavoprotein, cytochromes, pump out H pluses, produce the electrochemical gradient of H plus. Okay, ATP synthase will have them come in, okay, by chemiosmosis, okay, using the energy of the electrochemical gradient. They produce the ATP by oxidative phosphorylation. That's all you have to write, okay? So, and overall, guys, again, I don't know exactly the count here, guys. I mean, you probably have like 12 of these, 2 of that, guys. So you can kind of imagine how many ATPs you can produce, guys. 2.5 for NADH, 1 NADH, and 1.5 for FADH2. So you have an understanding of how many NADH, or how much ATP you can produce. They will never ask you how many do you produce. They'll not ask that question, okay? So I think one of the stuff that I went over with some of the folks here was that so this is AP might bring it up, whatever, So they might want to talk about, your teacher, I don't know, might want to discuss something they call uncoupler. Okay. And so of oxidative phosphorylation, uncoupler. So dude, I'm teaching bio, but I don't like these terms, uncoupling or coupling, So what does it mean by like coupling? Like where's the coupling process here? So according to textbook, right? According to bio, electron transport chain. So they refer electron transport chain what? Up to here. Up to here is electron transport chain. They pump out these pluses. That's ETC. Okay. So ETC is coupled with what? ATP synthase, ATP synthesis. It's just a strange thing. Okay? These two guys are coupled. They go together. Okay? One cannot, like, this cannot, like, you can't do this without this guy. They're coupled reaction. Okay? A coupled process, not a reaction. So they say ETC and ATP synthesis is what, guys? It's coupled by what? Electrochemical gradient of H plus. Okay? Because resultant of this guy is what? Electrochemical gradient of H plus. Okay? So the question is, what uncouples them? What, like, get rid of them? Is what, guys? So these two are tied by what, guys? Greater concentration of H plus here. Electrochemical gradient of H plus. Right? How do you uncouple it, guys? Yeah, like, okay, so H plus comes in through ATP synthesis, right? ATP synthase. So uncoupling is what, guys? Okay. Produce a channel where what? H plus can come back without using this guy. Okay, that's called uncoupling. You understand what I'm saying? So, yeah. So this is some of the stuff. Yeah. So, yeah, there is something we call uncoupler. It's called dinitrophenol. Dinitrophenol. That's an uncoupler for ETC and ATP synthesis. Okay, uncoupling is what? Yeah, yeah, dude, we're doing this, but what? Okay, no ATP synthesis. Okay, it's called, so this dinitrophenol. Okay, they punch a hole in there, and what? H plus comes through here instead of going through there. Okay, so yeah, questions for you guys. I have some of the questions to in the books here. Okay, who would intentionally use this? Yeah, who's gonna intentionally use dinitrophenol? Yeah, who's gonna use it? What do you think? Who's gonna use dinitrophenol, okay, to uncouple ETC and what ATP synthesis? What do you think? Yeah, who would use it? What do you think?
Muscle cell?
Muscle cell? Why would they do that?
To make it.
So you gotta understand, when they uncouple, what happens then? They're using up the glucose, but there's no ATP production. Okay? Who would intentionally will do this? Which means they want to utilize glucose, but there's no energy production. Okay? Which means what? Your body will respond to produce what? Respond to what? Burn up more glucose. Who will want to take this medicine of dinitrophenol? What do you think, guys? Yeah, well, yeah.
If you have too much glucose, like diabetic, maybe?
What is the problem with diabetic folks? Do you know?
Too little insulin.
So now with the problem with diabetes is what?
Insulin isn't active enough.
So there's type 1 and type 2. Okay, type 1 is what? They're not able to produce proper insulin because of damage that is done, or they're born with what? Not able to produce insulin at all, or insulin is not like appropriate insulin. There's something happened, okay? So there's a type 1. So if you have a type 1 diabetes, what happens then? What's the problem? The glucose in your blood will not enter the cell. That's a problem, okay? They cannot enter the cell at all. So type 2 is what? Your body produces insulin, but your body produces insulin for what? Cells are not absorbing them at a rate, appreciable rate. Okay? So typically this will not help them because what? The cells are not entering. The problem is because what? The glucose is not going into a cell. That's a problem. Okay? This happens inside the cell. So who would want to do this? What do you think? Yeah, what do you think, Daniel? DK, what do you think? Yeah, what do you think? Anyone?
Apoptosis? I have no idea.
Yeah. This dinitrophenol is what, guys? This is someone, who has this is a medication for obesity. So, obesity. Think about it, guys. So if you want to lose weight, they take this. Why? Yeah, dude, they're uncoupling this, okay? Your body will burn more glucose because why? Okay, they are supposed to, if I put like 1 glucose, I'm supposed to get 36 ATP. Okay, but I got 12 ATP. What is your body gonna do? Put more glucose. Do you know what I'm saying? Okay, so dinitrophenol is what, guys? This is something they utilize a lot in weight loss programs and medications. Just, so it's something that you can understand, right? I mean, with your teacher, I don't know if this is not part of it. I mean, they can definitely give you stuff like this, okay, on the APFRQs, pre-responsive. Okay, they'll explain it, what it does, and they will want to explain how does it work. Okay. Any questions, guys? And this is like how it works, guys, by the way, guys, and it's not a lot of time, but all right, let's take a look at, guys, the photosynthesis now, okay? I tell you guys one thing. A lot of folks are pre-med. You know what really pushed them away from being pre-med guys? The first year bio classes. So, you know, the first like the intro bio level class guys, when you guys go to college, that will take care of half of the pre-med semester. You know, so you know, so which means like it's hard to pass those classes, by the way. But if you know what the hell's happening, guys, before you go in, it will help you a lot. So you know, especially like the Olympiad stuff, man. It's above and beyond for the intro level bio class, by the way. So it's like it'll be a piece of cake for some of the folks. All right, so let's discuss, guys, photosynthesis. So I'm gonna pretty quick, guys. I gotta go over the form. We'll see, guys. Okay. Photosynthesis, guys. So this one, photosynthesis, guys. So we talked about this a little bit, guys. Okay, so this is sort of the opposite, right? Which are they're using the CO2. Okay, they're. I'm sorry, guys. It's not for me, guys. Can't do it. If I get too hot, guys, like I don't know. I think my brain enzyme like denatures or something. I can't think, guys. And the music yesterday, oh my god, like they're at 1 a.m. they're starting to play again. Like I was about to go to the Bronx actually, and then I was calling like the 311, like get on the call with them, and like I don't know. Yeah, it's but I got some sleep though. But dude, I have to. I don't like the summertime actually. But yeah, photosynthesis, guys. So you know, so basically it's the opposite process where you're grabbing a CO2. I mean, so very high level, right? CO2 turns into what, guys? Into glucose. That's what they will say, right? CO2 to glucose. That's what's gonna happen. That's what we know. So the process that this can happen, guys, where this can happen is what? Kelvin cycle. So we talked about this a little bit. Kelvin cycle, right? Kelvin cycle. So a CO2 goes in, okay? CO2 goes in, and what comes out? Yeah, what comes out, guys? G3P. G3P, okay? Glycerol 3 phosphate. This is Kelvin cycle. You understand, guys? Kelvin cycle is this, okay? Yeah, 3 CO2 goes in, and what? 1 G3P comes out. That's Kelvin cycle. So, yeah, CO2 to G3P, okay? This is what, from looking from here to here is what, guys? This is reduction process, reduction. Yeah, so we went from G3P to CO2, right? That's the Krebs cycle and what, ETC and all that stuff, guys. That's what? Oxidation, they take the electron out and what? ETC, okay? You're going backward. This process is what? Like reverse process of like, not like exact reverse, but the reverse process of what, guys? Of the aerobic respiration process, right? So what do you have to do, guys? You gotta put ATP for this. Also what, guys? Electrons, okay? So electron is what? Yeah, electron, right? So electron is given for the process, okay? Which is what? Like in here, they use what? They use NADPH, okay? So it's exactly the same as NADH except for what? They got one more phosphate group on them. One more phosphate, that's it, okay? So NADPH turns into what, guys? NAD P plus, H plus, 2 electrons will be given, okay? So they have to utilize what, guys? 6 ATP, okay? 6 NADPHs, okay? And what? Here they need 2 ATP here also as well. But 3 ATP, I'm sorry. So yeah, think about it, guys. To produce 1 G3P, glycerol 3 phosphate, they need to have what, guys? 6 ATP, okay? Or like 9 ATP. On a good day, and what? 6 NADPH, which is what? This is actually what? 12 electrons. 12 high energy electrons, okay? It's a lot of stuff that's needed for this process, okay? So CO2, guys, will go in, okay? So I mean, if you're like really interested in bio, like if you really like these stuff for whatever reason, okay? Whatever, like your brain, you got hit by something, you know? And then, oh my God, I mean, I want to learn just like, you know, more today. Oh my God, you know, hit your head somewhere. Oh yeah, so keep in mind, guys, here, just so you know, so we talked about it, right? There's enzyme that does it, which is Rubisco, right? Rubisco enzyme. That's the enzyme that will fix CO2s, right? Okay. So Rubisco enzyme will put CO2s with what they call, guys, ribulose, okay, biphosphate. Okay, ribulose biphosphate. Okay, and then so if you're dude, if you're interested in these stuff, okay. So they turn this into, so basically when Rubisco puts more CO2s on the ribulose biphosphate, okay, this is this they call RUBP. Okay, ribulose biphosphate, okay? Rubisco is what? Ribulose biphosphate carboxylase, that's what Rubisco stands for, okay? They turn this into, okay? Again, if you're interested in this, okay? Phosphoglycerate. Phosphoglycerate is what, guys? It's an intermediate of the glycolysis. So they're literally going what? They're just literally going backward of the glycolysis process. Okay. So energy payoff. Okay. They're going backward. Okay. So and then ATP. What happens, guys? Biphosphoglycerate. Okay. That's they're literally going backward. And then turns into what? G3P. Okay. Yeah. So glycolysis was what, guys? G3P. Okay. And then what? Biphosphoglycerate, and what? Phosphoglycerate. And you got to know that, okay? But they're literally going backward here. You don't know. Use ATP and ADHs and what? And later produce G3P. Okay? So, and this can what? Turn into glucose, right? After more steps and stuff like that. But now, that's Calvin cycle. Okay? There's nothing more we have to know about Calvin cycle, that's it. Okay? So, I told you we discussed about the 3 steps. Carbon fixation, reduction, okay, and what? The step of what? Regeneration, okay? So regeneration, that's the 3rd step. Fixation, reduction, regeneration, okay? Those are the steps, guys, of the Calvin cycle, okay? So now to do this, what happens, guys? You need to have what? ATP and ADPH. Okay, where are they gonna get it from is the question to ask yourself, right? So that's when we start to talk about the light-dependent reaction. Okay, LDR, light-dependent reaction. Light-dependent reaction of the photosynthesis will provide them with the ATP and what? Inert electron, which is high energy electron of the NADPH. Okay, you have to know this guy and those guys, okay, for photosynthesis. So now they ask more about this one than that guy, by the way. So LDR. So keep in mind, guys, here, so this happens, so this is a chloroplast, right? Chloroplast is where the organelle where this can happen, okay? And now, chloroplast has what, guys? Something they call thylakoid, okay? Thylakoid, right? Thylakoid is where they will be able to run the process, guys, by the way. Listen carefully, okay? Calvin's cycle occurs what, guys? In the stroma, okay, portion of the chloroplast, okay? And now, the LDR happens what, guys? On the thylakoid, okay, membrane of the chloroplast. Okay, they asked the question, by the way. Okay? They're asking, oh, where does it happen? And blah, 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 Especially stuff like this, right? So the acetyl-CoA formation of the mitochondria, for example, where does it occur? Matrix of the mitochondria. They ask this question because, that's AP. So now the question is, guys, how do they produce ATP and ADPH?
So to make the G3P, it's the 6 and the 6. But then to replenish, it's the 9 and the 6.
Yeah, so during the regeneration phase, they need about 3 ATPs. Yeah, so overall, it's 9 ATPs needed for one cycle. They will never ask that. AP, they don't care. Back in time, when I was in the AP, they're like, how many? One cycle. They ask those questions. But nowadays, it's very unlikely unless your teacher I think so. If they talk about it.
Got it.
Yeah. I mean, if you really know this process and you want to show off, then you can write that on the AP. But if you make a mistake, they're gonna make it wrong. So oh yeah, you're writing all this crazy stuff and you make a mistake for writing on not needed steps but you got it wrong. So might as well not talk about it if you really don't know 100%. All right, so let's do the reaction, guys. So this one, guys, So it occurs at a thylakoid, you guys. So if you look at this, but this is the membrane of thylakoid, okay? So let's be very simple about it. I gotta finish this in a few minutes, guys. So, so they have something called the photosystem, okay? The photosystem is a system, right? There's a bunch of crap in there, okay? So the photosystem has, you know, like the reaction center, you know, which has the pigments, okay, such as chlorophyll, okay? And then they have electron acceptor, blah, 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 you know. And now they have PQ, cytochrome, PC, and then they have another photosystem. And then, so I'm going to be pretty quick here, guys. So they have FAD, I think. They have reductase. Yeah, so this is basically, guys, what you have to know for the overall what happens, guys, for the production of ATP. Okay? And then they have, I'm sorry, I forgot to put, they got ATP synthase. So let's take a look, okay? What's gonna happen is, so what will happen is, okay, so the fundamental understanding is all the same, okay? They need 9 ATP, right? How do they get it, guys? ATP synthesis, okay? ATP synthase, same thing. Just like how we talked about, guys, for the mitochondria ATP synthesis, guys, exact same thing, okay? They produce a concentration greater than H plus. The ATP synthase will what? Have the H plus, like, you know, go out by chemiosmosis, and what? Yeah, produce ATP by what? Chemiosmosis. Same thing, exact same thing, but it's a little different, guys, okay? So what happens is, so these photosystem, okay, this is photosystem 1, okay? They got, so for us, for now, okay? They got something we call chlorophylls. Chlorophyll. Okay, chlorophyll is pigment, right? So chlorophyll is good at what? Absorbing some band of light. Okay, so some of the light that we get, guys, they can absorb it. Okay, especially, guys, around 700 nanometers, which is a red color. Okay, red color. So these guys are pretty good at absorbing around like 680 or like, you know, I might have swapped these guys, by the way, but around there, which is red light. So, you know, like basically what will happen is chlorophyll can absorb a wave of light, okay? And what the hell do they do, guys, here? So chlorophyll, okay, has a magnesium center, okay? It looks very similar to like hemoglobin, by the way, okay? So having a metal in the middle, guys, what happens, guys? Sunlight will hit it. It can what? Excite the electron of magnesium, okay? So it doesn't have to be magnesium, but it's around the magnesium, guys, that can excite the electrons. So what happens, guys? The electron gets excited, it gets accepted by electron acceptor, and what? Being able to run. So electron gets excited, what happens, guys? Being able to run. Electron transfer chain, again, same thing, okay? So, you know, but now, the difference between ETC of the mitochondria versus ETC of this guy as well, what's the difference, guys? The difference is, the high energy electron, okay, for mitochondria came from what? Carbon of the glucose. What about this one? It's coming from the chlorophyll that's excited by the sunlight. That's the only difference, okay? So the electron will be handed down to plastoquinone. That's just carrier electron. Not that you will never bring these up because you know your teacher will probably don't know. They probably don't want to bring it up because you know, okay? So it will be handed down to cytochrome. Cytochrome does exactly the same thing. What do they do, guys? They pump the electron, the H plus, okay? But yeah, listen carefully. So for mitochondria, what do they do, guys? They pump out from the in to the out, okay? It's the opposite here. They pump in the H plus into the thylakoid space, okay? That's the difference. So they're not pumping out, they're pumping in. That's the difference. And then they hand the electron to the PC, a plastocyanin, okay? And then what? These guys don't pump anything, by the way, okay? And the electron will be handed down to the second photosystem, okay? And the sunlight will be shining on the photosystem 1. What happens, guys? Electron gets excited. So keep in mind, guys, here, just like how we talked about, guys, something needs to replenish the electron. Okay, like, dude, they don't have unlimited electrons, right? Like, dude, do they have like unlimited electron, guys? Like, oh yeah, here we go, here we go, here. They can't do that. What do they do, guys? They have to get it from somewhere, right? They have to replenish, right? Where do they get the electron from? Yeah, where do they get it from?
ADP.
Huh?
ADPH.
What?
ADP.
NADPH?
Yeah.
Make sure I'm saying. NADPH, right? The electron in this guy. What do they do? What do they do with the electron from this guy? They have to give that to a carbon for glucose for production of G3P. They can't put the electron back into this guy because this guy will be going to Calvin cycle and give electron to carbon. Okay, so no. Which means what? There gotta be something else, right? Water, H2O. Water. So you have plant. You heard it, right? Like, yeah. If you have plants, okay, how do they survive? Like, you got plants, right? Like, what do? You put sunlight and what? Water, they grow themselves, okay? Why water? You gotta ask yourself, like, why the hell do plants need water? Water will do this. Electron gets replenished by water, okay? So, again, like, oh yeah, like your dad, oh my God, you know, I need to put some water. Well, they got no water, you know? Why? Because what? Water will give them electrons. You want a lot of stuff today, okay? You can tell your parents, okay? But this guy told me, water gives electrons. Your dad, you know why you need water for your plants? Oh yeah, you're getting electrons. I knew that before. So yeah, that's where the electron from, guys. You know. So and then the O2 goes out. Okay? So O2 is what, guys? So what they call a byproduct of what? Of the photosynthesis, right? And then what happens, guys? Yeah, the electron gets replenished. So what will happen is now, so yeah, they like again the H plus concentration build up inside the dielectric. What happens, guys? They will leave through the ATP synthase, chemiosmosis, and produce ATP by what? By phosphorylation. And they call this phosphorylation what? What do they call this, guys? Photophosphorylation. They call this. They call this, guys, photophosphorylation. Okay, photo. They don't call this. Yeah, they don't call this oxidative phosphorylation. They call this what? Photo. Even though it's the same thing. Because what? They use sunlight. Dude, I tell you, man, I don't like bio, but I mean, dude, bio is not my favorite subject, by the way. I don't like it. It's like they're not. I don't know. Yeah. So photophosphorylation. Okay. So the way they produce ATP is what? Photophosphorylation. And then and then what happens here? Keep in mind, guys. Okay. Yeah. If you think about this, right? They need more ATP than what? Like 6 NADHs. Which means, like, dude, they need more ATP. Okay, so ATP is like, it's more in demand for them, you know. So which means what, guys? Okay, so what happens is, if they just follow this way, okay, and then they'll go FAD, okay, and reductase here. What happens, what, guys? Yeah, this is where electron gets excited again. So yeah, this way forward, there's no pumping of the H pluses, okay? It's over. So keep in mind, guys, the electron gets excited here. What happens, guys? So reductase what, guys? Yeah, what do they do, guys? The enzyme NADP plus 2 electron turns into what, guys? Into NADPH. That's what's gonna happen. Okay, so they use the energy of the sunlight. They pump the electron into a higher energy state, and what? Yeah, they what? Produce NADPH for Calvin cycle. Okay, that's what happens for the second excitation. So from here on, what happens, guys? Yeah, pumping is over. Okay, this yeah, you probably listen carefully. This is what they call non-cyclic or cyclic, whatever. Okay, electron transfer chain, non-cyclic, because there's no cycle, just one way. But now, yeah, what will happen? Okay, so this process is a little bit more sophisticated. Okay, yeah, they need more ATP. So I'll explain why they need more ATP. A lot of times ATP is like more short-handed, okay? So like water is available, but ATP, man, it's so hard to get. What do I do, guys? There is you gotta know this, guys, a cyclic, they can make this into a cycle. So listen carefully, okay? So electron gets handed down, it goes down, H plus pumps in. What happens, guys? So from here, this is a non-cyclic. This sequence is non-cyclic, right? But electron here can be passed down back into the cytochrome. Okay, as electron here, it goes back to this guy. I mean, so it goes to FAD and it goes back, but whatever. Okay, so it goes back to cytochrome. Okay, and then what? Oh yeah, which means what? They use the energy of the electron from high energy. What? They what? They pump the H plus more, which can what? Produce greater electrochemical gradient for what? More production of ATP. So this right here is what they call a cyclic portion of the electron transport chain for chloroplast. Okay, they run this process to what, guys? To the extent of what? Yeah, if they until what? They need more ATP. They run the cyclic process. Okay, so keep it. So there's no cyclic process in oncology, but they do have it, as we know. So because why? They have a two-photo system here. So if enough ATP was produced, what happens, guys? Yeah, okay, they go into non-cyclic now. So you turn, and then go down to this way, because we know. So again, what's the purpose, guys? Produce ATP using ATP synthase, and also what? Produce what? NADPH for the Calvin cycle. Okay. I guarantee you guys they'll ask this question on the AP. They ask a lot of questions like this. I mean, they ask about the absorbency, but I'm not going to talk about it right now. Okay? We don't have time for that. Any questions, anyways? Questions? All right, so two more things to talk about, okay, right now. So that is, one of them is, guys, photo respiration. Okay, I'm going to talk about it in about 5 minutes. And the second one, guys, is C4 plants and CAN plant. Okay, that's the second one, and that's it. And next week, guys, we'll talk about the cell cycle and the cell communication. All right, so and one thing you have to ask yourself, right? Plants 500 million years ago, they live in water. Okay, they live on top of the water. So you've probably seen some of the plants live on top of the water, right? Like what? Some flowers, right? Like live on top of water. So for them, right? They got water, right? And they have what, guys? They got the sunlight, right? Perfect combination, right? Sunlight and water, right? Why do they have to come to land? Okay, why do they have to come to land, right? Like, what's the purpose of? So there's more plant and land than water, okay? Right now, okay. And then one of the plants that's really dominant is called the angiosperms. Okay, the flowering plant. Okay, why did they have to come to land? That's something you have to ask yourself. Like, what the heck? Why? 500 million years ago, why did they decide to come to land? Why did we decide to come to land? Yeah, that's something that we can ask ourselves. You know. All right, so let's talk about, guys. This is very important, guys. Photo respiration. You have to know what the hell happens here, okay? Like what's going on here? Like, okay, why do we like have some different plants and stuff like that, guys? Okay. Photo respiration. Okay. So there's a little problem, okay, for the plants. Okay. So now, the problem is with the Calvin cycle. So there again, there's RUBP, okay, ribulose biphosphate. There is what? CO2 must go in, and there's a job of the ribisco, which is ribulose biphosphate carboxylase. And what do they do, guys? They produce what? They produce something we call glyceraldehyde. I'm sorry, phosphoglycerate. Glycerate is what? 3-carbon molecule. Okay, this is just what we talked about, yeah? Phosphoglycerate. So, so guys, ribisco, CO2, RDT, glycerate. So, to keep in mind, guys, every plant will need to do this. If they're running photosynthesis, they gotta do this. They need to have this enzyme. Okay? But we talked about this a little bit, okay? When temperature gets a little hotter, when temperature goes above, like, I don't know, like 35 degrees Celsius or something, okay? Temperature gets a lot hotter, what happens, guys? There's a big problem for the ribisco. Ribisco has one job only. I'm telling you, ribisco has one job. They got one job, okay? Just like you guys have to study. You guys got one job. Study, okay? Your mom sends you here, you study, you know, get 100 on the test. But you guys can't even do that. Some of you guys can't even do that, right? Some of you guys, no, I'm not gonna do it. And yeah, they get to be 70. Okay? Just like Rubisco messes it up, right? Instead of bringing a CO2, when temperature goes up, what happens, guys? They bring O2 gas. That's a big problem, guys. Instead of bringing a CO2, they got one job. What happens, guys? They're bringing O2 gas. Okay? You understand what I'm saying? Yeah, so like that, yeah, you think about it, guys, okay? If you put an O2 gas in there, are you going to get G3P? You gonna get it, guys? What do you think? Absolutely not, right? You're not, dude, like what the hell? Like, they think they're gonna get this, they're getting something else, okay? I think they get glycolate, that's what they get. So, they're not getting it, okay? So what do they do, guys? So when they bring an O2 instead, they're not getting this, they're getting something else. Okay. So what do they do with this? So what the hell do they have to do? They have to go extra step, okay, to put this back into RVP. Okay. They gotta go here, okay, and then what? They have to go into a different cycle to get RVP, which is really biphosphate, to run it again. Okay, because they put the wrong stuff in there. So this is an expense of ATP. They use ATP for this. Okay, so you know, they use ATP for this to turn it back in there. So you know, so this is a big problem. Okay, so you know, for the plants. So this right here is what they call, guys, photo respiration. Okay, so you know, because they're bringing the O2 instead of CO2. That's called a photo respiration. They use what? Like, dude, this, remember I said on a good day. Okay, this occurs at a 9 ATP. But if in case the O2 goes in, what happens, guys? Yeah, this is what happens. They use more ATP. So, you know, so this is a problem, guys. For some plants, okay, when temperature gets a little hotter, what happens, guys? It becomes very inefficient. Okay, the Calvin cycle becomes very inefficient for them. Okay, so if they're not getting a CO2, what happens, guys? They're gonna keep open the CO2. They're gonna keep open the stomate. They're gonna lose more water. They're gonna lose more ATP. So the things are gonna wear out. They're gonna die. That's a problem with what we call, guys, photo respiration. Okay, so your teacher's not gonna go too crazy here. By the way, you have to explain what it is. I mean, so instead of bringing a CO2, they bring an O2 gas in there. So they might ask you about why they do this. Okay, because what? Temperature gets a little hotter, the configuration of the enzyme might change a little bit. Okay, and keep in mind, guys, this plant, you know, Rubisco is one of the most, like, I don't know, man, very ancient enzyme. Okay, back in the primitive atmosphere, way back. Okay, we didn't have a lot of O2 gases in the atmosphere. Okay, because we have so much O2 gases now, then this becomes a problem. Okay. So yeah, what do you think, guys? Do you think plants will die now because of this process of photo respiration, guys? A lot of plants will die because of this, right? Do you think plants will die? Now they evolved, become what? They got a different plant. They got C4 plant. Okay, so listen carefully, okay? Plants, they can only do this process, or the fixation process. What do they call this? They call it what? They call this C3 plant. Okay, they call it C3 plant. They call it C3 plant because why? The first thing they produce when the carbon is getting fixed is glyceraldehyde, which is a 3-carbon molecule. They call it C3 plants. But now, there's something called C4 plant. Okay? Which is what, guys? Like this, so they have a different way to fix our CO2. And that is like this. So you gotta at least know C3 plant, guys, okay?
C3 or C4?
C4. So C4 plant, guys, they have a mechanism, okay? So they have a mechanism, guys, okay? This is the outside. This is the inside of the plant. So this cell is called a mesophyll cell. Okay, mesophyll. This is called a bundle sheath cell. So you have to you gotta have a good understanding of what's happening here, okay? So mesophyll cell, which have exposure to the outside, they do not use Rubisco. Rubisco is like, okay, so you're garbage, so I'm using a different enzyme. Okay? So there's use an enzyme called PP. Okay, they use an enzyme called PP carboxylase. Okay, so PP stands for phosphoenol pyruvate. Okay, so PP, guys, PP, right? So they have a PP, So this is also one of the steps of the glycolysis, just so you know, okay? Phosphoenol pyruvate, okay? So PEP will fix a CO2 by PEP carboxylase, okay? And it produces something that you should already know, which is oxaloacetate. Do you have to know this, guys? I don't think so, okay? You might know the enzyme, PEP carboxylase, okay? Rubisco versus what? PEP carboxylase, okay? And they produce what? Oxaloacetate, okay? And this will, they will turn this into a malate, okay? And then they unload a CO2 and turn into a pyruvate. So, and then all the expensive ATP, they turn it back into PP. So, basically, yeah, what's happening? So, there are two cells working together. Mesophyll cell and what? The bundle sheath. So, they use a different enzyme to fix a CO2 and what? Hand the CO2 to what? A cell that's going to run the Calvin cycle. Okay, so they capture the CO2 and they give the CO2 to what? A cell underneath them, bundle sheath, and they're going to run what? Calvin cycle with the enzyme Rubisco. So Rubisco has no exposure to the outside, okay? They're what? Being handed down a CO2. Okay, that's what we call C4 plant. Why is C4, guys? Because why? Oxaloacetate is what? 4-carbon molecule, that's why, okay? So, and then, yeah, the CO2 will be given, guys, to the bundle sheath, and they're gonna run the Calvin cycle, and what? Being able to produce G3P. So now, why? So some plants will run this, such as what? Corn plant, okay? And now they do this at expense of ATP. They use ATP for this. Yeah, do ATP. They use the energy for this, okay? So on a good day, so where do they get it from? Light-dependent reaction. Okay, so keep that in mind, guys. So this is what they call, guys, a C4 plant. Okay, they have a different mechanism of gaining the CO2 gas. Okay? Any questions, guys? Questions, concerns, let me know.